This video is part 2 for subtopic 3.2 of chromosomes. So here are a list of understandings that we'll cover, and we'll basically cover the latter half of these understandings related to diploid and haploid nuclei and karyograms, and then the applications and skills that will be related to this video, as well as a bit of guidance. So we'll start with haploid nuclei. So recall from the previous video, we ended with talking about homologous chromosomes. Homologous chromosomes have same number of genes and the same types of genes, but they differ in their alleles. Our nuclei can be divided or characterized as being haploid or diploid. So haploid nuclei will contain one chromosome of each type. And remember, a typical nucleus will contain two chromosomes of each type. So a haploid nuclei will typically contain half the number of chromosomes, but they'll have one chromosome of each type. And we represent that as N. And so in humans, there are 23 chromosomes. So to represent this haploid number in humans, we would simply write N equals 23. And this would indicate that in a human cell, there are 23 chromosomes. Now, there are cells where we find one chromosome of each type, and these are the gamete cells. So sperm and egg cells contain one chromosome of each type. And remember that when fertilization occurs, so if you have an egg and a sperm that each have 23 chromosomes, so 23, and when fertilization occurs and you get fusion of sperm and egg cell, this will form a cell now that has a total of 46 chromosomes. So this is where the 46 chromosomes come from, 23 from dad, 23 from mom, to give us a grand total of 46 chromosomes. And those 46 chromosomes are then in that first cell that's produced, then go on to divide and divide and divide to produce that organism, which can grow into an adult to produce its own gamete cells and continue that circle of life. So these sperm and egg cells are said to have a haploid nuclei because they have one type of each chromosome. In haploid nuclei, chromosomes will split at the centromeres during anaphase to give us our two sister chromatids. And then chromatids will move to opposite poles of the cells to become chromosomes in these newly created cells. In a diploid nuclei, this is where we have two chromosomes of each type, and we represent that as 2N. So remember from the previous slide, we stated that one chromosome will come from the mother, this is the egg cell, and the other will come from the father, this is the sperm cell. So in humans, we have 46 chromosomes. So to indicate this diploid nuclei, we would write that as 2, that's a 2, 2N is equal to 46. Now, a diploid nuclei will come together when haploid gametes fuse together during reproduction to produce a zygote with that diploid nucleus now. This cell will divide by mitosis, producing more diploid nuclei. So cell division through mitosis will produce more diploid cells. So cells that undergo mitosis are going to produce more cells that have a diploid nuclei. Now, each cell has two copies of every gene other than those on the sex chromosomes. Remember that for each chromosome, there's two of each type, one from dad, one from mom. And so those chromosomes will have a copy of every gene between them. So there'll be two copies of each gene present. except for the sex chromosomes. And remember, those are going to be different chromosomes. So the sex chromosomes are going to be defined as X chromosome and Y chromosome. And because they're different, they're going to have different genes on them. With chromosome numbers, the chromosome number is a characteristic feature of that particular species. A chromosome number does not indicate how complicated an organism might be. Organisms with a different number of chromosomes are unlikely to interbreed. So in order to generate a successful offspring, organisms have to have similar chromosome numbers. If there's a mismatch in chromosome numbers, then you do not get proper fertilization that occurs. The number of chromosomes can change during the evolution of a species. So if chromosomes fuse together, the overall chromosome number will decrease. If chromosomes split, the chromosome number will increase. During human evolution, it's thought that two ape chromosomes fuse together to produce the human chromosome 2. So in humans, chromosome number 2 looks like a very long version of two chromosomes from a chimp that fuse together. So there is a different chromosome number between humans and chimps, and why you could never crossbreed chimps with humans. When comparing chromosome numbers, you have to keep in mind that some organisms have a few large chromosomes and others have many small chromosomes. All eukaryotes have at least 2 different types of chromosomes, so the diploid chromosome count is at least 4. So if we look at this horse threadworm, it has the bare number of chromosomes required. And then here you can see that the chromosome numbers are always going to be even. You'll never have an odd number of chromosomes. If there should be 2 of each chromosome, then you'll always end up with an even number. Or you should always have an even number. Now, chromosomes will play a role in sex determination. So as mentioned before, there are 2 chromosomes in humans that determine sex. There is the X chromosome, and we've talked about this previously, I believe, that is large and has a centromere near the middle. So, the X chromosome, typically when viewed under a microscope or electron microscopy, has like a X shape to it. The Y chromosome is smaller and has a centromere near the end. So it typically has like these little runty ends here and kind of a longer end. Because the X and the Y determine sex, they are called sex chromosomes. All other chromosomes are referred to as autosomes. And so the combination of chromosomes that you get determines the gender of the individual, the biological gender of the individual. So if you end up with two X chromosomes, you are female. If you have an X and a Y chromosome, you are male. So the X chromosome has many genes that are essential in both males and females. It controls the development for both. So all humans have at least one X chromosome. The Y chromosome, because it is so small, only has a small number of genes with it. So only a small part of the Y chromosome has the genes as a small part of the X chromosome. So there's a little bit of overlap. The other genes on the Y chromosome are not found on the X chromosome and are not needed for female development. So looking at the Y chromosome a little bit closer, so the Y chromosome has a series of genes called the sex-determining region of the Y chromosome, or the testes determining factor genes. These are found on the Y chromosome, and it tells you that you are basically going to be male. So it develops, helps develop testes by allowing for testosterone production. So it encodes the production of testosterone, which develops male sex characteristics. A fetus with two X chromosomes does not have the testes determining gene, and so does not produce testosterone, and so through the progesterone secreted by the mother, will develop ovaries, and as a result, will produce their own female sex hormones. So when sperm and egg fuse together, obviously sperm is from dad, egg is from mom, who determines what the sex of the baby will be? So as I mentioned before, females have two X chromosomes, and therefore they will pass on one of each of their two X's in each egg. Remember that an egg is a gamete cell, so we'll only have one of each type of chromosome. So if a female typically has two X chromosomes in a cell, that egg cell, when it fully develops, will only have one X chromosome. And so if you think about a male, then the same situation applies to males. So males are XY, they have one of each, they have two of the sex chromosomes, and so when sperm develop, they will only have one of these sex chromosomes. So you'll have one sperm that contains a Y chromosome, and a second sperm that will have that X chromosome. So the possible combinations are depicted here in terms of the sex chromosomes contained within sperm cells. So you have one with an X, one sperm cell with a Y. In terms of the egg cells will always contain an X chromosome since they're from mom. So if a sperm cell containing an X chromosome fuses with that egg, you get two X chromosomes and thus a female fetus resulting. If that sperm contains a Y chromosome and fuses with that egg, you get that combination of XY, resulting in a male fetus. So basically, the gender is going to be determined by the chromosome carried in the sperm. So half those sperm contain X chromosomes, and the other half contain Y chromosomes. So daughters inherit their father's X chromosomes, and sons inherit his Y chromosome. So it is the father that determines the sex of the baby. So in terms of visualizing the complete set of chromosomes within an organism, this is done through karyograms. So a karyogram is going to show the chromosomes of an organism in those homologous pairs in decreasing length. So a karyogram will take all those chromosomes, it will pair up the homologous chromosomes, the ones with the same gene sequences, same lengths, and a centromere in the same position, and order them from longest all the way down to shortest with the sex chromosomes at the end. Now, the position of the centromere and the pattern of banding allows chromosomes that are of a different type but similar size to be distinguished. So the banding pattern, the position of the centromere, and the length are all distinguishing features. Now, from the karyogram, you can determine a karyotype. So the karyogram is basically an image of the chromosomes of an organism arranged in those homologous pairs of decreasing length, so that's that picture. And then from the karyotype, that's derived from the karyogram, this is basically the property of an organism. So the karyotype will show the number of types of chromosomes in an organism. The karyotype is studied by looking at karyograms. It determines if an individual has all 46 chromosomes, has two X chromosomes, or has 46 chromosomes and has a combination of XY. And it also determines if an individual has any chromosome abnormalities. So chromosome abnormalities can result in various genetic diseases. So these changes in chromosome numbers can have effects on the individual. So when you have monosomy, which is the loss of a chromosome, And in this case, the most common one is a genetic disease referred to as Turner syndrome. This is where the individual just has an X chromosome, but no Y chromosome or no second X chromosome. So these individuals are usually short, have webbed neck, and are sexually underdeveloped. The more common type of chromosome number change is that of trisomy. This is the gain of an extra chromosome. This results in basically 3 of one type of chromosome. So, for example, there is Down syndrome, which has a trisomy for chromosome 21, which can result in mental disabilities. There is Edwards syndrome, which has a trisomy for chromosome 18, so 3 copies of chromosome 18, which results in intellectual and physical disabilities. There's like Pautel, which is a trisomy on chromosome 13. And then you can get trisomies for the sex chromosomes as well. So, for example, there's Klinefelter syndrome, where there are 2 X's and 1 Y chromosome. There's Jacob syndrome, where there is an X chromosome and 2 Y chromosomes. There is what's called the triple X syndrome, where there are 3 copies of the X chromosome. So this is the end of the video for part 2 of 3.2.